Discovering FishBase and SeaLifeBase, an in-depth introduction. Welcome to this introductory chapter on FishBase and SeaLifeBase. In this video, you will be given an overview of FishBase and SeaLifeBase, including their history, coverage, interfaces, and applications. This video is accompanied by a set of self-assessment questions that will help gauge your understanding of this chapter. Let's begin. What is FishBase? FishBase is a premier global biodiversity information system on finfishes. It is publicly available online, and free to access. It is available through 9 mirror sites and in over 20 languages. And it receives around 700,000 visits per month. At present, FishBase covers over 35,800 fish species, compiled from about 63,000 references. It also features almost 330,300 common names and nearly 65,100 pictures. The database has around 2,550 collaborators. What is SeaLifeBase? SeaLifeBase is a global online information system on all marine species in the world, apart from finfish. Like FishBase, it is publicly available online, and free to access. It is available through 3 mirror sites in over 20 languages, and garners around 500,000 visits a month. SeaLifeBase currently covers nearly 71,800 species, compiled from over 40,400 references, and is in partnership with about 370 collaborators. The database contains almost 60,100 common names and 15,700 pictures. FishBase and SeaLifeBase both offer online information with full source attribution, data for ecological and ecosystem-based models, standardized data for interoperability and tools, and serve as a resource for education and capacity building. Quantitative Aquatics or Aquatics is the home of FishBase and SeaLifeBase. Aquatics is a non-profit organization based in the Philippines. It is part of the FishBase Consortium, which is composed of 15 highly reputed academic and research organizations across 6 continents. The FishBase Consortium provides scientific and technical guidance to ensure that FishBase and SeaLifeBase remain current and relevant with evolving issues on biodiversity, environmental recovery and protection, sustainable development, fisheries management, and climate change. In over 30 years of continuous operation and development, FishBase has become one of the largest and most extensively accessed online public resources in the history of scientific research, and is in the top 1% of all cited items published in this century and the previous century. A new study that measured the scientific impact of FishBase reveals it is also one of the most highly cited databases. According to the citation analysis, FishBase has received over 10,000 citations from hundreds of peer-reviewed journals in Scopus from 1994 to 2020. While Google Scholar attributed nearly 15,000 total citations to FishBase for the same period, with an average of over 1,200 citations per year from 2017 to 2021. FishBase and SeaLifeBase are widely used throughout the world. Regions that use FishBase and SeaLifeBase the most are Europe, the United States, Brazil, and Australia. Both of these species information systems are also indispensable resources for developing countries. As the leading providers of freely accessible online global aquatic biodiversity information, encompassing both fish and non-fish species, FishBase and SeaLifeBase have received acknowledgments and honors from various entities. FishBase has received the ISI Current Web Content Award and the Study Sphere Award of Excellence. In 2004, FishBase received both the Study Web Academic Excellence Award and the CGIAR Science Award for Outstanding Scientific Support Team. While SeaLifeBase was commended in Linksgiving.com's Weekly Link Award Hall of Fame 2008. In 2017, the Fisheries Society of the British Isles bestowed the Lacren Medal, which recognizes the lifelong contributions made by the FishBase Consortium and the entire FishBase team to the field of fish biology and fisheries science, with a focus on conservation, training, or public understanding of the discipline. FishBase and SeaLifeBase share these awards with their partners, collaborators, and supporters. The history of FishBase and SeaLifeBase. It all started with a box of index cards full of data and two brilliant minds meeting to create what has become the world's premier encyclopedia on fish. FishBase was started in 1989 through the collaboration of ECLARM, now World Fish, and FAO, with funding from the European Commission. Back then, topics and tools in FishBase focused on trophic ecology, distribution, population dynamics, ecology, and taxonomy. But the coverage has expanded over time. FishBase milestones. FishBase was first made available in CD and DVD format in 1995. And was made available on the Internet in 1998. In 2008, FishBase hit 30,000 species, or 90% of all known fish species in the world. In 1994, Daniel Pauly moved to the University of British Columbia in Vancouver. And in 1995, The Sea Around Us was born through the collaboration of UBC and Pew Charitable Trusts. The Sea Around Us is a research initiative that assesses the impact of fisheries on the marine ecosystems of the world and offers mitigating solutions to a range of stakeholders. In 2001, Deng Palomares joined The Sea Around Us to assure that fish-based data was used in The Sea Around Us and that The Sea Around Us results were also used in FishBase. In 2000, the FishBase Consortium was formed to provide continuing support for FishBase work. Now with 15 members, the FishBase Consortium acts as the scientific advisory committee of FishBase and SeaLifeBase. In the same year, Rainer Froese left the Philippines to work at the Guiomar Helmholtz Center for Ocean Research in Germany. In 2004, FishBase adapted the relative environmental suitability model by Kristen Kashner to develop AquaMaps. AquaMaps are computer-generated, predicted global distribution maps for marine species. AquaMaps for fish species were first made available online in 2005, featuring the native distribution for a given species and its modeled year 2100 native range under the IPCC-A2 emission scenario. In 2022, under the leadership of Gabriel Regondo of the University of Miami, AquaMaps moved toward the development of a new species distribution ensemble modeling framework called AquaX. AquaX employs multi-model algorithms that quantify the current and future global distribution of marine species at high spatial resolutions and assess model performance and uncertainties. Back at UBC, the idea of having a FishBase-like information system for non-fish aquatic organisms was brought up. Nicholas Bahi wrote the concept note for a database that would hold non-fish aquatic organisms. In 2005, with funding from the Oak Foundation and under the leadership of Deng Palomares of The Sea Around Us, SeaLifeBase was born. SeaLifeBase milestones. SeaLifeBase was launched online in 2008 and reached 13,000 hits per month during the first 6 months. During the same year, Sea Around Us, FishBase, and SeaLifeBase collaborated to deliver an Ecopath with Ecosym training. In 2010, SeaLifeBase documented the marine biodiversity of the South China Sea, logging almost 50,000 species with mandatory information. This activity strengthened the partnership between FishBase and SeaLifeBase and marked the shift in the database's strategy for documenting the marine biodiversity of the world, by working on one marine ecosystem at a time until all 66 large marine ecosystems are well covered. In 2014, The Sea Around Us started generating species distribution polygons. Country data from The Sea Around Us, FishBase, and SeaLifeBase was harmonized to provide the best species map via AquaMaps, and used as the basis of its distribution polygons. In 2015, the reconstruction of Philippine fisheries was conducted by the SeaLifeBase team under the leadership of Deng Palomares. In 2018, the experience of SeaLifeBase in reconstructions and database development was instrumental in assisting The Sea Around Us with the 2016 Global Catch Update with the support of Min.de.ru funding. And from 2021 up to the present, FishBase, SeaLifeBase, AquaMaps, and The Sea Around Us continue to work together to provide information and services, this time under the EcoScope project on ecocentric management for sustainable fisheries and healthy marine ecosystems. FishBase and SeaLifeBase are huge repositories of information on all marine species in the world contained in interrelational databases. Here, we present the data coverage, the data quality standards we follow, and how this data can be accessed on the FishBase and SeaLifeBase websites. Let us now explore these databases together. The first step is to identify the species, and that starts with the name. Taxonomy is the primary way basic information about plants and animals is organized, stored, and classified. Here we show the general biological taxonomic ranking system. In FishBase, it covers class to species level only because all species are under phylum Chordata, while in SeaLifeBase, it covers phylum to species levels of classification given the different non-fish marine species groups. We recognize the importance of getting scientific names right because information attached to the wrong species creates misinformation. Thus, each database follows a taxonomic backbone. For FishBase, we use Eschmeyer's Catalog of Fishes, and in SeaLifeBase, we use the World Register of Marine Species. The classification and names of species in both databases are further validated by experts, internally by a scientific advisor and taxonomic director, and externally by recognized authorities on species. The synonyms table is where we store the scientific names given and used for a species. This is used to properly assign a piece of information and helps keep track of the changes or confusion between names used in a reference, information, and locality, among others. It is also essential for studying biodiversity and serves a huge purpose, from conservation management to inventories, trade, and identifying threatened species. Each synonym record provides the scientific name, the etymology of the name, the status whether it is accepted or a synonym, the status details, the reference used for that synonym and a comment field for other taxonomic information about it. Lastly, we have one of the most important attributes for this category, the common names. These are the names commonly used for a species in a locality, in a particular dialect, or even as a trade name used commercially. This allows our users to find a species just by using what they know and how that species is called locally. Most of the database users access the species data using common names. Common names have also been used for other applications, like creating infographics for specific projects. Here are some examples presenting how common names were used to link to their known commercial names and corresponding scientific names. Learn about where a species can be found. In our database, the distribution of a species is assigned according to FAO area, ecosystem, country, and occurrence, along with data on its areas of introduction. Let's take the example of the thornback ray, Raja clavata. It is a fish species found in the northeast Atlantic, within FAO Area 27. Specifically in the North Sea. In the United Kingdom. In the Thames estuary. This displays the different coverage of distribution in the databases from the largest, the FAO area, to the smallest, showing the exact occurrence point where a species can be found. In the country, ecosystem and FAO area tables, we further classify their presence in that area as native, endemic, stray, introduced, reintroduced, extirpated, questionable, in cases where an occurrence needs further confirmation, and misidentification, for records that are known to have incorrect identification, or an error in assignment to a locality due to other factors. In this example, you can see the countries listed for Raja clavata and based on the list, they were assigned to the native distribution, with one record for a questionable occurrence in Madeira Island. In the country table, we also have source qualifiers, checklist, map, occurrence, and region. These are used as indicators of validity based on the references used. The most reliable and our top priority sources are checklists, followed by map distributions, occurrences that provide an exact point where they are found, and lastly, the general region where they are found. The general region is used only if there is no other reference available confirming its presence in the area. Other information provided in the country and ecosystem tables includes environment, whether freshwater, marine, or brackish, the known abundance, commercial importance, aquaculture, regulations, live trade, use, as game or bait, and threatened status in that country. The occurrence table provides more specific information, starting from the reference used, name used in the reference, catalog number, locality, and country. Other information available are the occurrence points, latitude and longitude, depth range, year of recorded occurrence, date, time, sex, size, habitat where collected, gear used, collector and identifier, etc. We also have an introduction table that pertains to introduced species. This example shows the introductions of the fish Anguilla anguilla with information on the year it was introduced, area of origin, area of introduction, status of establishment, and ecological effects. Now let's learn where and how a species lives. We have a variety of tables to capture this. For this section, let's take examples from Sepia officinalis. Let's start with the ecology table that provides the detailed habitat, vertical zones, substrates, and associations. We also have the food items, a list of foods that the species consumes, and the diet table, which provide information on what the species eats at a specific locality. These are useful in assessing its ecological function and impact for the construction of ecosystem models. Additional tables involving these data are the food consumption and ration tables, for data at the individual and population-based levels. Lastly, is the predators table, that lists reported predators of that species. Now let's learn how species grow. In the database, information on population dynamics is divided into a series of tables. Growth is a process through which size, based on length or weight, changes with time. The growth table contains information such as asymptotic length, the growth coefficient K, and natural mortality. Maximum size presents information on the maximum length and weight, and maximum age in the wild by locality. The largest value from this table is also used in the maximum size data for the species in general. Length weight provides parameters that can be used to estimate the biomass of a species in a locality based on length observations. Data for length-length relations are also available because species are measured in different length types according to their shape. Some species also have data for length frequency and recruitment. We also have tables for reproduction and life history, that provide information on maturity, spawning, eggs, broodstock, fry nursery, larva, larval dynamics and larval speed. Here you can see one of the information products developed based on reproduction and life history information. It is a poster showing the true scale size at maturity of selected commercial fish in a particular locality that helps fishers and buyers see the minimum size at which a fish can be sustainably harvested and bought. Other species information available include morphology, physiology, including metabolism, disease, vision, sounds, etc., genetics, aquaculture, and nutrients, among others. We also have supporting information on species. First are the species identification keys, which provide in-depth information in terms of classification, morphology, and distinguishing characteristics. This is supplemented by pictures that give a face to the animals we provide information for, and aid in species identification. All information in the database is properly cited, with complete information for each reference. Each species has a bibliographic record that gives you the information source, title and author, the species name used in that reference, the page number, and what data was encoded from that reference for that species. For more information on each of the topics presented here, you can refer to the FishBase book through the link found at the upper left portion of the species search page. This opens to links to the different chapters in the book, FishBase Concepts, Design and Data Sources. The book is also available in French and Portuguese. How are FishBase and SeaLifeBase useful? In this segment, you will learn more about the online interfaces of FishBase and SeaLifeBase, covering the species summary page as well as the different tools available for both practical and advanced uses. You will also learn about examples of large data applications and collaborations using FishBase and SeaLifeBase. Let's begin. On the web, information for a species is presented through the species summary page in FishBase and SeaLifeBase. Let's dive into its key sections and learn how to access various information about a particular species. On your browser, call the species search page using either www.fishbase.org for fishes, or www.sealifebase.org for non-fishes. You can do a species search using either the common name or the scientific name. Type the common name in the common name field, for instance, tuna, or sea turtle, then click search. A list of results matching your query will be displayed. Look for your species of interest and click on its name. Alternatively, if you know the scientific name, type it in the genus and species fields or the genus plus species field. In our example, we type Tunis thinus in FishBase and Chelonia midas in SeaLifeBase, then click search. This returns the corresponding species summary page. This page provides a comprehensive overview of the fish species in terms of taxonomy, environment, distribution, morphology, biology, life history, and more. Now, let's take a closer look at its key sections. At the top of the page, you'll find the scientific and common names of the species, along with its taxonomy. This information helps you identify the specific fish species you're interested in. FishBase also provides species photos and illustrations that can assist in accurate species identification, especially for non-experts. AquaMaps is also integrated in FishBase, which can be used for species distribution analysis, habitat suitability, climate change impact assessments, and fisheries management. Scrolling down, you'll see a section dedicated to the environment and ecology with information on the habitat, depth range and climate zone where a species is found, followed by the section on a species' geographic distribution, with information on countries, FAO areas and ecosystems where they occur, as well as access to georeferenced species data, and the section on length, weight, and age of the species. Moving further down, we come to the section with a short description on the meristics, morphology, and morphometrics of the species. This is followed by the section on the biology of the species, and the section on life cycle and behavior, including information on maturity, reproduction and spawning. Identification of species, scientific research, conservation efforts, fisheries management, and stock assessment all depend on this information. A section on the main reference used for the species is also provided, while other sources of information are likewise referenced throughout the species summary page. Now, let's scroll down and explore the species conservation through human uses sections. Here, you'll find information on a species' IUCN Red List conservation and threat status, as well as its evaluation under the Convention on International Trade in Endangered Species or CITES, and the Convention on the Conservation of Migratory Species or CMS. This information can contribute to conservation efforts and help raise awareness about endangered fish species. There is also information on whether a species poses a threat to humans. And a section on human uses, providing information about the species' economic importance to fisheries, aquaculture and game fishing, with links to other sources for production and landing statistics, species profiles, and stock assessments. These data are valuable for fisheries management and aquaculture planning. Further down the page, you'll find the More Information section. The listed topics link to data derived from scientific studies such as estimates of maximum size, growth rates, natural mortality, diet, reproduction, population dynamics, and many more. Information is also available by country, ecosystem, and FAO fisheries area. This is followed by the section on tools that includes a biodiversity quiz, an e-book, field guide and identification keys for the species, wizards for length frequency analyses and generating life history parameter estimates. Special reports feature links to other websites, for instance on species fact sheets. There is also an XML facility for downloading species information like the summary page, point data, common names and photos of the species. There are also links to other Internet resources, which are helpful if you want to learn more about a particular species or explore relevant research articles and publications. Finally, at the bottom of the page is the section on estimates based on models, which includes the mean and range of the preferred temperature of a species, phylogenetic diversity index or uniqueness of a species, Bayesian length-weight A and B values based on length-weight relationship estimates, trophic level from diet studies, generation time from growth studies, resilience or population doubling time, prior R estimates from stock assessments, fishing vulnerability or intrinsic extinction vulnerabilities to fishing, vulnerability to climate change, X-vessel price reliability, and micronutrient concentrations from fish nutrient studies. We have just given you a general overview of the FishBase and SeaLifeBase species summary pages as comprehensive resources for anyone interested in information on fish and non-fish species. Up next, we will show you examples of practical as well as advanced uses of these databases. There are a number of tools in FishBase and SeaLifeBase that are of practical use for students, researchers, and those involved in marine biodiversity conservation and monitoring. In this section, we will be showing you a few of these tools, what they are, where to find them online, and how to use them for your work. Learn more about the Match Names tool, Species Identification tools, Analytical Graphs, and Information Gaps tool. The Match Names tool allows you to compare a list of scientific names against FishBase and SeaLifeBase to get the current valid name of the listed species. It also helps in identifying duplicate species or synonyms, misspellings, or wrong entries on your list. The tool is found in the lower part of the FishBase and SeaLifeBase search pages. Scroll down to the Tool section and click on the Match Names button. In the box, you can type the genus and species names as shown in the example. Or cut and paste from a spreadsheet or text document. And then click Submit. This returns a list of results from matching names against FishBase. We see that, based on our example, 28 scientific names were submitted. In the returned matched species table, 12 species have corresponding species in FishBase. The matched species table shows the species names from your data, the corresponding valid scientific name and author, and the species family, order, and class. Note that submitted names that are different from valid names are marked with a yellow background. Record number 7 shows that the submitted data is a misspelling of Onko Ring Kus Cha Wit Sha. Whereas, record number 11 shows the correct spelling of the species name as N. gralis japonicus. For users of our FishBase, the spec code can be used to link the species list to related species information in various tables in our FishBase. Scroll down to the Unmatched Species table at the bottom of the page. Listed are the submitted scientific names with no matches in FishBase. This would typically include names of non-fish species, or invalid entries, or non-scientific names such as Ingralis spp, Elasmobranchs or tuna. We see that 16 species names from our example have no matching names in FishBase. In this case, they refer to non-fish species and could be inspected using the corresponding match name tool in SeaLifeBase. In the returned results, we see that, based on our example, all 16 scientific names that were submitted have corresponding species in SeaLifeBase. We note however that records number 11 Baryteuthis magister and number 15 Gonadus septemdentatus are synonyms of the species Baryteuthis magister. Thus, the list contains a total of 15 unique species. The matched names results can then be used to prepare a definitive table of species for a report or publication. Or to perform advanced queries in the R FishBase and R AquaMaps packages in R, that provide programmed access to data for thousands of species in the FishBase, SeaLifeBase, and AquaMaps repositories. We have just shown you the Match Names tools. Next, you will learn more about the FishBase identification tools. There are a number of identification tools in FishBase that anyone can use to identify fishes they have caught on fishing trips or seen while snorkeling. These tools make use of the huge number of fish photos compiled in FishBase, along with species distribution and morphological data. To identify a species, you can choose from among 3 identification tools. These 3 tools use different approaches to identification. One uses identification keys, another uses morphometric measurements, and a third tool uses pictograms and photos. The approach that uses pictograms and photos is the most convenient to use and suits most users. Let's see how it works. On the FishBase search page, scroll down to the Tools section and click on Quick Identification button. FishBase uses pictograms to help users identify the family to which their fish belongs. They are grouped into 6 classes: ray-finned fishes, sharks and rays, chimeras, lobed-finned fishes, lampreys, and hagfishes, each accompanied by a description of species belonging to each group. If, for example, you are trying to identify a fish that looks like a herring you found in France, the tool will ask you to select an area and country from the drop-down lists, and to select an ocean area, if you are trying to identify a marine or brackish fish, or a continent for freshwater fish. For our example, we will select the Northeast Atlantic to specify the ocean, and France for the country. That is the first step. Next, click on the pictogram where you see groups of fishes that look like a herring. In this case, we choose the group ray-finned fishes. This returns a set of pictograms grouped by the different orders of ray-finned fishes. Scroll down the page and click on herrings. On the succeeding screen, you can find the pictogram for species belong to families of herrings reported from France in the Northeast Atlantic. Click on Clupiidae. This is the family to which herrings, shads, sardines and menhaden belong. The tool then narrows down the possible species to the Atlantic herring Clupea haringus, and the European sprat Spratus spratus. You can click on the photo or the name to see more information in FishBase to help verify your identification. But, sometimes, identifying a species from several photos can be difficult. The Quick Identification tool offers additional options to help narrow down the species photos to choose from. At the top portion of the page, you will see fields that allow you to apply additional filters. For instance, by specifying a genus, or including species occurring above a specified depth limit. If you have a specimen on hand, you can also specify characteristics such as the total number of spines in the dorsal or anal fin, or enter the total length of the species or its maximum body depth before clicking Redo. If you want to try out the other identification tools in FishBase, you can click on the links at the bottom of the page to access tools for identifying species through identification keys or by morphometrics. These tools, however, are best suited for advanced users. We have just demonstrated how you can use FishBase to identify fish. In the following section, you will learn more about the different analytical graphs using FishBase and your own data. The Analytical Graphs tool allows you to generate a number of graphs using data currently in FishBase. There are 9 graphs in this tool. The axiometric graph illustrates the empirical relationship between the von Bertalanffy growth parameters k and asymptotic length of species, or L infinity, which enables analyses of many growth parameters and comparisons among species of widely different shapes. Axiometric graphs can be used for analyses that look into the difference in growth performance, such as this study done for captive versus free-living Nile tilapia. Here, the axiometric graph illustrates that the growth performance index of fish in aquaculture is much higher than for conspecifics in open water. It can also be used to test life history hypotheses such as this study that predicted fish species in Greek seas would generally be smaller in size compared to populations elsewhere due to the oligotrophic environment in Greek waters. The M versus K graph shows the relationship between natural mortality, M, or the proportion of fishes dying from natural causes such as predation, diseases, natural disasters and old age, and the growth coefficient, K. Here, we see that K is a good predictor of M, which confirms that K is among the predictors influencing M. The LM versus L infinity graph plots length at first maturity versus asymptotic length. The estimation of LM requires substantial data and is usually not available for many species. An examination of the link between LM and L infinity shows there is an empirical relationship that can facilitate the estimation of LM from L infinity. Here, we see empirical relationships identified for females and males from 265 fish species. The M versus L infinity graph shows natural mortality against asymptotic length. It can be used to illustrate the influence of size and environmental temperature on natural mortality. Here, the M versus L infinity graph shows how natural mortality differs between species in waters below 20 degrees Celsius and those in waters above 20 degrees Celsius, which are typical of tropical fishes. The length-weight graph plots A and B values of the length-weight relationship W equals A times L raised to B. A plot of log A over B forms a straight line for most species. Thus, estimates that are clearly below or above that regression line are possibly questionable. There are a few other lesser-known graphs. The reproductive load graph shows reproductive load against the asymptotic length. This can be used for evaluating hypotheses on growth and reproduction in fishes. The relative brain weight graph illustrates the relationship of brain weight to body weight. Fish have evolved the brain size they need, so the difference in brain size among species may be used to draw inferences on their niche. The gill area graph relates gill area and body weight. Hypotheses on fish growth can be tested using gill area. Gill area measurements are of practical use in pollution and ecotoxicological studies. Lastly, the relative oxygen consumption graph compares respiration rate and body weight, one of the factors affecting metabolic rate. It is useful in comparative physiology and in solving problems in rearing or transporting live fish. You will find these graphs in the Tools section of the search page. Move down the first column and click on the Graphs button. Multi-species graphs can be generated on demand according to 8 categories: family, class, climate, order, habitat, salinity, ocean, and inland waters. Choose a category, then make a selection using the drop-down list provided. For our example, let's click on the Family button. Select a family by choosing from an alphabetical listing of families or a list of family common names. Or simply start typing an entry, and the tool will automatically search and fill the rest of the field for you. In this case, typing cods will bring up cods and haddocks. Click on this to make the selection. In this example, we will choose the M versus K graph. This graph illustrates the empirical relationship between natural mortality, M, and the growth coefficient, K, based on records for species of the cod family Gattidae, in red dots, against those for species of other families in FishBase, in yellow dots. Fields found below the graph allow you to add your own estimate to the plot and compare it with other estimates. For instance, we plug in an M of 0.5 and a K of 0.4 and click Redraw. A black dot representing our estimate is now added to the redrawn graph, and we see that this estimate is within the range of estimates reported for cods. For more information about the M versus K graph, click on the About This Graph link found on the upper left-hand side of the graph. Note that the Add Estimate fields are found in all the analytical graph tools, and that users can add and redraw a plot one estimate at a time. So far, we have shown you examples of the analytical graphs generated by family. But you can also call graphs by order, class, climate, habitat, salinity, ocean, and inland waters. Finally, these graphs are also available for selected species and can be accessed from the More Information pages in the Species Summary page, under topics like maturity, length, weight, and growth. Here you can view the different graphs and the data behind them, which can be copied to a spreadsheet for your own analysis. We have just finished guiding you through the different analytical graphs in FishBase. Up next, learn about the Information Gaps tool, which is practical for finding out which kinds of information are not yet available in FishBase or SeaLifeBase for a given group of species. We are now going to explore an incredibly useful tool in FishBase called the Information Gaps tool. With the use of this tool, you will be able to identify fish species for which there is missing or incomplete information, allowing you to contribute to the collective scientific knowledge. Let's get started. From the FishBase homepage, scroll down to the Tools section, and go to the Information Gaps tool and click on the radio button. It's a powerful feature that allows experts, scientists, researchers, and enthusiasts to contribute to filling in the missing pieces of information about a particular fish species. Once you're on the Information Gaps page, you'll see a search bar and a list of various information categories related to fish species. Let's examine some of the key categories in more detail. First, we have country. This category focuses on species that are present in a certain country. Alternatively, users can make a selection by taxa, in particular by genus name, which can be selected from the drop-down list. Next, go to the List to Output category and select one of 8 options: all fishes, fisheries, aquaculture, threatened, aquarium, bait, game fish, or all important species. Next, move to the Environment category, and select the preferred habitat of a species, such as fresh, for freshwater, saltwater, for marine, or all for both environments. Suppose we select all marine fish in Greece. Let's now view the report. The list of species will be displayed according to the options we specified. The species status, life history, reproduction, ecology, feeding habits, and more are all covered in the Information Gap report. For each topic, a plus sign indicates that there is information available, while a minus sign means that information is missing. Notes at the bottom of the list provide more details about the table. Just above the Notes section are the summary statistics per topic, giving the number of species with available data and their percentage of the total species in the list. In our example, out of the 544 marine fishes in Greece, only 155 species or 28% have information on mortality while 93% have species photos. Let's now talk about how you can contribute to filling in the information gaps. For example, let's select all fisheries species in Greece. A list of fish species that lack information in that category will then be presented. Clicking on a specific species will take you to its species summary page, where you can make a request to add or edit information by sending your comments and/or suggestions through the user feedback link at the top of the page, featuring contact options via email under Comments AMP, corrections, or through our Facebook page. Depending on the category, you may need to gather scientific research papers, field observations, or other credible sources to fill the gaps. Once you've submitted your contribution, the FishBase team will review the information before it is included in the database. Together, we can create a valuable resource for researchers, scientists, and fish enthusiasts. In the previous section, we presented to you tools in FishBase that are of practical use. In this section, we will introduce tools for advanced or power users of this database. You will learn more about the Life History tool, Trophic Ecology, and the Ecopath Parameters tool. Let's begin. Thousands of species of fish are used by humans for food, sports, the aquarium trade, or are threatened by overfishing, pollution, environmental degradation, and climate change. However, life history parameters such as growth and size at first maturity, which are important for management, are known for only a fraction of these species. The Life History tool was created to provide estimates with error margins of important life history parameters for all fishes. It also enables users to generate estimates using their own data. Let's go back to the FishBase species search page and look up the surmullet, Mulus surmulatus. On the species summary page, scroll down to the Tools section and click on Life History Tool. This page presents a combination of published data and estimates of important life history parameters for all fishes. These include parameters on population dynamics such as maximum length, the growth coefficient K, natural mortality M, length at maturity and length-weight relationships, parameters on reproduction such as fecundity and yield per recruit, trophic level and food consumption, resilience or productivity, exploitation, and a few other parameters. Definitions of these parameters are provided through the About This page link at the upper left-hand corner of the page. Note that the estimations are based on default values from FishBase and so may not be applicable to all populations. For this reason, the Life History tool was designed to be interactive. You can enter your own data and recalculate the estimates. The estimates are presented along with error margins, and you can see how your own data compares. Let's use the surmullet for our example. Let's say, you find out from your surveys that the L-infinity of this fish in your area is 40 centimeters, but you also need an estimate of the length at which you could expect this fish to reach maturity. You need to know the length at which this fish could potentially be spawning. You can use your L-infinity as input in the tool to get an estimate of LM, the length at which your fish would be mature. Simply replace the default L-infinity value with your data of 40 centimeters total length, and click on Recalculate, and the tool recomputes length at maturity to be around 22.9 centimeters for your fish. Note that when changing L-infinity, several other life history parameters are recalculated as well. Another interactive feature of this tool is the estimation of fish weight from its length. In the length weight section, you can enter the length of your specimens and you get an estimate of its weight. For example, let's replace the length 40 centimeters with 35 centimeters, and retain the measurement to be that of total length. Then click Recalculate. Note that the weight now changes from 870 grams to 570 grams wet weight. You can also recalculate the weight using your own estimate of A or B. And use these in combination with estimates of A and B derived from other studies, which can be accessed through the length weight data link. As you can see, estimates can be recalculated for several other parameters on the Life History Tools page. The data points used to calculate estimates for several parameters in the tool are available for download, or can be cut and pasted, so you can examine them if you wish or use them outside of the tool. Have a look and see how you might be able to get initial estimates of the parameters you need for your own research. Good luck exploring the tool. Let us now look at the Trophic Ecology tool found on the FishBase and SeaLifeBase search page. Trophic Ecology examines how organisms obtain energy and nutrients and how these transfers of energy and nutrients shape the structure and dynamics of ecosystems. It explores the feeding relationships between producers, consumers, and decomposers. FishBase and SeaLifeBase provide trophic ecology data classified under diet, food items, and predators. FishBase additionally covers food consumption and ration. So, how do we access trophic ecology data? In the FishBase or SeaLifeBase website, scroll down to Information by topic and choose from the options listed underneath Trophic Ecology for the topic you're looking for. Accessing data this way provides you with a list of all species with available data based on your chosen topic. You can also look up species-specific trophic ecology data. Let's find diet information for the green sea turtle, Colonia mitas, as an example. After typing in the species in the SeaLifeBase search field, you will be redirected to its species page. Scroll down to more information and select diet. This will open up the diet composition page of the species. Selecting a link from the main food column will redirect you to the summary of diet information from a particular reference. The diet compositions page shows the percentages, in weight or volume, of the different types of food items reported from various diet studies. The first three columns, main food, percent and trophic level, refer to the general classification of the food item based on our standardized hierarchy, the percent weight or volume contributed by a food item to the stomach content of the reported species, and the estimated trophic level of the species' diet composition, respectively. Country and locality refer to the area where the species diet study was conducted. Data presented in this page can be sorted either alphabetically or in order by toggling the arrows beside the column headers. Selecting any link from the main food column will open up the food and feeding habits summary page, providing additional information such as sample size or the number of specimens, the mean length of the specimens in the sample in centimeters, coupled with a length type field, percentage of empty stomachs in the sample, period where the known feeding months of the species are indicated, the diet percent, showing a detailed breakdown of each food item, adding up to 100%. Food 2 and 3, showing standardized hierarchies that describe food items with increasing levels of detail. The prey stage, part field refers to the food item's life stage when it was consumed. Item name specifies the food consumed in more detail. The comments field is used for additional information that further explains or describes particular food items. Let's go back to the more information section of the species summary page for Colonia mitas. Click on Food Items. The Food Items page highlights food items documented in the species' stomach contents or are otherwise known to be ingested by the species. The first three columns, Food 1, Food 2 and Food 3, are standardized hierarchies to describe the food items in increasing levels of detail, while the last three columns, Food Name, Country and Predator Stage, describe the scientific name of the food item, if specified, the country where the food item was recorded, and the species' life stage when it consumed its meal. Selecting any link from the Food 3 column will open up the food item details page, providing additional information such as the food group field, which refers to the family or higher order group of a food item, the prey stage part field, which refers to the food item's life stage when it was consumed, the commonness field, which refers to the percentage of specimens containing the food item, and the remarks field, which provides other pertinent information, including but not limited to localities, dates, and methods. Once again, let's go back to the More Information section and click on Predators. The Predators page lists reported predators of a particular species. The four columns describe the country where the predator was recorded, the standardized hierarchy of functional groups describing the predator in specific detail, the family of the predator, and the scientific or common name of the predator. Selecting any link from the name column will open up the predator details page, providing additional information such as the locality field, which refers to the site where the study was taken, the predator stage field, which refers to the predator's life stage, the prey stage field, which refers to the prey's life stage when it was consumed, and the percent of stomach contents field, which lists the percentage the prey contributes to the stomach content of the predator. Let's go back to the information by topic section, this time on the FishBase search page. We will examine the three other topics on trophic ecology that are currently only found in FishBase: species by food items, food consumption, and ration. Species by food item presents a list of species with food item data that can be filtered according to the database's Level 3 standardized food item hierarchy, covering 60 functional food groups. Select any functional group from the drop-down box at the top of the page and click Submit to filter for species that consume your selected food group. For example, let's select bony fish and click Submit. In the returned list, select a species in the first column, for instance, Alepicephalus rostratus further down on the list. This will redirect you to the food items page for this species. Back at Trophic Ecology under the Information by Topic section, select Food Consumption to get a list of species with population-based estimates of food consumption. Click on a species name, say, the Atlantic herring, Clupea haringus on the list. This will redirect you to the food consumption list for this species, where the columns provide information on the following. Country refers to where the study was conducted. QB refers to food consumption per unit biomass per year. W infinity refers to the asymptotic weight in grams of the von Bertalanffy growth parameter. K refers to the growth coefficient of the von Bertalanffy growth parameter in years. Mortality refers to the mortality of juveniles and adults used for simulating age-structured populations. Temperature refers to the annual mean water temperature for the stock in question. Food type refers to the food consumed in the quantity indicated by the QB value. And reference links you to the source of the data. Selecting any link from the QB column will open up the population food consumption details page, providing additional information including the maintenance QB field, which refers to maintenance food consumption per unit biomass per year. The T0 field, which refers to age at length 0 of the von Bertalanffy growth parameter, in years. The salinity field, which refers to the mean salinity regime to which the stock is exposed throughout the year. The locality field, which refers to the site where the study was undertaken. And the remarks field, which provides other pertinent information. Let's return to the Trophic Ecology section under the Information by Topic section, and look at food rations. This presents a list of species with estimates of daily food consumption by fish of a specific size and its related parameters based on ration studies. Click on a species name. Here, let's click on the Atlantic cod, Gadus morhua. This redirects us to the ration list of the species. The columns provide information for the following. Weight refers to the total wet weight of individual fish, in grams. Ration refers to the percentage of the ratio between the weight of the total food consumed in a day by individual fish to its wet body weight. K1 refers to gross conversion efficiency, which is the growth in weight or the weight of food ingested over a given period. Evacuation rate refers to the fraction of stomach content passed through the hindgut per hour. Temperature refers to water temperature conditions at the time of sampling or experimentation. Salinity refers to the mean salinity regime to which the stock is exposed throughout the year, and Food 1 refers to the general functional group that the species consumes. Selecting any link from the weight column will open up the ration and related information page, providing additional data such as the locality and country fields, which refers to the site where the study was done. Food 2 and food name fields, which specify food consumed with more detail, and the comments field, which provides further descriptions of length or weight measurements. Data below the comments field is part of the methods used section, which lists methods used for the estimation of daily ration. Other pertinent details may also be added under additional remarks. Hopefully this increases your understanding of the trophic ecology tables in FishBase and SeaLifeBase. By analyzing this data, researchers can monitor species and their habitats, assess the effects of human activities, develop sustainable management strategies, and other potential applications to real-world decision-making and conservation efforts. The consequence of collecting data is its use in data-driven actions. To mention a few specific applications, data on food items can be used to identify staple food preferences in various species for which detailed diet composition data are not available, and to make a preliminary estimate of trophic level. The information compiled in the predators table is useful to fishery and conservation workers, as predator-prey relationships may help explain the status of some fish stocks. The information is also used for the construction of trophic pyramids. Finally, data from trophic ecology can be used for higher-order analyses using other information from FishBase or SeaLifeBase. By combining diet data with the relative sizes of prey and predators, one can generate a graph that shows a trend supporting the hypothesis that the ratios of predator and prey sizes are similar among fish of different species. Understanding those relations provides key insights that can help facilitate strategies for predation dynamics, resource management, ecosystem conservation, and health status. The Ecopath modeling approach involves gathering data on the different components of an ecosystem, such as species composition, biomass, and feeding habits. These data are then used to construct a mathematical model that represents the flow of energy and nutrients between the different species in the ecosystem. The Ecosystem Parameters tool in FishBase draws from the database's tables on a species' population dynamics, life history, trophic ecology, and environment and consolidates key data that is accessible through a single table. In the FishBase search page, the Ecopath Parameters tool can be found in 3 sections: information by country or island, information by ecosystem, and the Tools section further down the page, offering different options for accessing Ecopath modeling input data. Going through information by country or island, select a country or island from the drop-down list, wait for the screen to refresh, then click on the Ecopath Parameters button. You will then be asked to select an FAO area to filter for inland or marine fishes. Under Information by Ecosystem, click on the drop-down list and choose from the listed categories of ecosystems, for instance, marine ecoregions. Again, the screen will refresh and display a second drop-down list. Choose an ecosystem from the list, for example, the Adriatic Sea. Then click on the Ecopath Parameters button. For the third option, under the Tools section, click on the Ecopath Parameters button to call the page Parameterization of Ecosystem Models. You will see an option to generate model input data for species by ecosystem with an optional filter by habitat type. Or by FAO area, for instance, let's choose the Atlantic Northeast. This is to be further filtered by a country selection. Here, we choose Germany. There is also an optional filter according to feeding type. We will leave this blank for our example, then proceed to click Submit. This will return a table of species for the selected area, with data for ecosystem modeling which include a species' maximum length, its trophic level, growth parameters, length-weight parameters, QB ratio or food consumption, food and diet data, predators, and depth data. A plus sign indicates that there are one or more data records available for the species. A minus sign indicates that there is currently no data record available for the species. The default sort of the list is by length of species from highest to lowest. Other options include sorting by trophic level, family, and habitat. This sorts this list by highest to lowest for trophic level, and alphabetically for family and habitat. Simply click the radio button beside each option to sort by your preference. Let's have a closer look at the underlying data, using the example of the Atlantic bluefin tuna Tunis thinus. Click on the plus sign under Growth over M column. This opens the table of growth parameters for Tunis thinus, based on 29 records for the species. The table presents data for parameters such as L infinity, the growth parameter K, mortality, and so on. There is also information such as the country and locality of the study, whether the data record is questionable, and based on a captive specimen or one caught from the wild. By default, questionable records are unselected. Therefore, only data records that are check-marked are used for the graphs available through this page. These include distributions that graph the data densities of L infinity, K, and mortality that allow the examination of the correlation of parameters log K and log L infinity. And axiometric graphs based on the species length and weight records, with values for the Atlantic bluefin tuna plotted in red. There are also links to other graphs found in the upper left portion of the page. Let's now click on the plus sign under LW. This opens to the length-weight parameters page for Tunis thinus, presenting length data. A, B, R squared, and other estimates. Study area, and so on. Doubtful records are also flagged. In addition, each record is weighted by scores. The higher the score, the better the data, while doubtful records get a score of 0. At the bottom of the data table, you will find the preliminary length-weight parameter estimates based on the selection of non-doubtful records and weighted by the scores. These include the number of studies used to compute the geometric mean of A, mean of B, and the standard deviations of log 10W, log A and B, as well as the preliminary length-weight relationship and a corresponding weight range at 95% confidence interval. The length field is editable. You can plug in a length measurement, and the corresponding weight and range are recomputed. For example, let's replace 172 centimeters with 150 centimeters and press Enter. This recalculates the corresponding weight to 54.7 kilograms, and a range of 40.4 to 74.1 kilograms. In the upper right section of the page, there is also a Show Graph link that calls a log 10A over B graph overlay based on the tabulated data. This can be used to identify and decide whether to exclude or give less weight to studies that are far from the regression line in the graph. Lastly, you can also incorporate more data using the Include Genus or Include Family options. Based on our example, this will return records including length-weight parameters for other Tunis species, or species under the Scombridae family, respectively. QB returns the food consumption list for Tunis thinus, which shows data from population-based food consumption estimates for the species. In our example, there is only one record of QB estimates. The record includes the country where the population was sampled, the food consumption per unit biomass, the asymptotic weight, the von Bertalanffy parameter K, the natural mortality, the annual mean water temperature since temperature affects growth and metabolism, the food type, which includes choices for the food items involved in the QB estimate, and the link to the corresponding reference for the data. Diet and food contains information on the diet composition of a species. A species record includes the country and locality where the diet study was undertaken based on a specific reference. The diet data includes the main food or main functional group of the food item, broadly grouped into detritus, plants, zooplankton, zoobenthos, and nekton. Percent refers to the median contribution of a food item to the stomach contents of fish, either by percent volume or percent weight. The trophic level is estimated from the diet composition and the trophs of the food items. Predator life stage refers to the life stage of the species when the diet composition study was undertaken. Finally, click under Predator to see the list of organisms that prey on a species. The resulting list shows the scientific names of the known predators, accompanied by details such as their family, functional group, and, if available, the country where the predation was observed. You have just completed the tour of the Ecopath parameters tool and tables. For more details on these tables, you can click on the More Info link at the upper right-hand corner of the page. The compilation of different kinds of species information at different spatial scales has made it possible for FishBase and SeaLifeBase to develop large data applications in collaboration with key project partners or in support of important collaborations. In this section, we briefly introduce some of these large data applications, including Ecopath, AquaMaps, the FishBase Guide App, Fish Online, and examples of knowledge products for biodiversity studies. Ecopath is one of three components of the Ecopath with Ecosum, a free and open-source ecosystem modeling software suite at ecopath.org. Ecopath is a powerful tool used to model ecosystems, helping researchers understand the flow of energy and matter through different components of the system. It provides a snapshot of the ecosystem's structure and function at a given point in time. The model is based on two master equations on production and consumption that represent trophically linked biomass pools such as single species or species groups in ecological guilds. The Ecopath model was initially developed at NOAA by Jeffrey Polovina in the early 1980s and has undergone further development for over more than 30 years through the works of notable fisheries scientists such as Carl Walters, Billy Christensen, and Daniel Pauly, and software engineers worldwide, from developing Ecopath 2 in the 1990s to the development of Ecopath with Ecosum into the 2000s, and the shift to centralized community-driven development in 2013 under the Ecopath International Initiative. Ecopath with Ecosum 6.7 is underway through the support of projects such as the EU Horizon 2020 EcoScope project and is planned for release in 2025. NOAA, in 2007, recognized Ecopath as one of the 10 most significant scientific breakthroughs in its 200-year history, citing how it has revolutionized scientists' ability worldwide to understand complex marine ecosystems. Models can come in the form of network indices and are used to assess the status of ecosystems based on a framework, such as the EU Marine Strategy Framework Directive, MSFD. MSFD defines 11 descriptors to achieve good environmental status, GES, a state that ensures the continued use of marine resources for generations to come. Ecopath outputs help form indices that measure GES descriptors, enabling scientists and other stakeholders to focus on areas in need of attention. Sample indices include 1. Detritivory over herbivory, D, H ratio. This ratio represents the relationship between the two food sources. In this case, knowing the aggregate trophic level of an ecosystem is a crucial factor in the computation of the ratio. 2. Fin Cycling Index, FCI. This ratio represents the percentage of energy flow being recycled in an ecosystem. Here, knowing the aggregate biomass of an ecosystem is needed in the computation of the ratio. 3. System Omnivory Index, SOI. This ratio represents the level of omnivory in an ecosystem. Knowing the feeding habits in an ecosystem is essential in the computation of the ratio. Ecopath also provides value in fisheries management, as it enables managers and key personnel to simulate various scenarios and examine their effect on ecosystem dynamics. Fisheries management models that use Ecopath can consider both direct and indirect effects of individual factors as they manipulate input parameters accordingly. Sample models include the following. 1. Stock assessment and sustainable yield estimation. By considering predator-prey relationships and food web dynamics, researchers can account for the role of species in estimating maximum sustainable yield, MSY, in a multi-species context. 2. Marine Protected Area, MPA, design. Ecopath can assist in identifying key areas for protection based on ecological importance and how that affects local fisheries and ecosystem recovery. And 3. Historical ecosystem reconstruction. By reconstructing past ecosystem states, researchers can identify historical baselines that can help set realistic management targets and better understand long-term changes that can occur in an ecosystem. As a key data source for Ecopath, FishBase strives to provide robust and reliable data to the fullest extent possible. These can be accessed through the Species Ecology Matrix and Ecopath Parameters tools found on the FishBase and SeaLifeBase web interfaces. Biodiversity studies have been done in FishBase and SeaLifeBase in collaboration with museums, the academe, and in support of conservation efforts for a country, region, continent, or ocean. Results of these studies are available on the species page under the Information by Country, Island or Ecosystem, on FishBase and SeaLifeBase search pages. This allows you to search species group by biodiversity status, such as endemicity, threat, and introduction. Using the species information page, we can find data and create different biodiversity maps. We can determine what species are there in a country, EEZ, marine ecoregion or LME. Predicted occurrence of these species are extracted as georeferenced points from AquaMaps. Diversity equations are used to compute for species diversity representations such as Simpson's and Shannon indices and range rarity. Species richness maps can be created on a global scale. It can be shown also by class, by order, and by family. We can also show species richness using areas of interest on a map. This one shows species richness in large marine ecosystems. And this one shows % probability of occurrence of species in an EEZ. Habitat preference index maps can also be shown using the Simpson's index formula. Also, range rarity can be computed to show the endemicity or rarity of species in a particular area or MPA. Biodiversity information is helpful in natural resource management, habitat protection and in species conservation. AquaMaps is a free, interactive online platform and tool for modeling the geographic distribution of marine species. The modeling approach was adapted from Kashner et al. 2006's Relative Environmental Suitability Model for mapping global distributions of marine mammals and adapted for fish and other aquatic species by FishBase and subsequently, by SeaLifeBase. AquaMaps combines species biological data and point data with environmental datasets to compute species-specific environmental preferences, and uses these to predict the potential distribution of various marine organisms in different regions of the world's oceans. The tool considers environmental factors such as depth, sea temperature, salinity, primary production, sea ice concentration, dissolved oxygen, and distance to land to generate distribution maps that show where a given species might be found under specific conditions in both the contemporary period and under the RCP 8.5 2050 climate scenario. FishBase and SeaLifeBase are primary sources of AquaMaps model input data such as the FAO areas where the species naturally occur, the bounding box limits of the species' native range, its habitat and depth range, as well as georeferenced species occurrence data. Currently, over 33,000 marine species have been mapped using data from FishBase and SeaLifeBase. These maps are available for individual species, illustrating its current native range, its predicted range by the year 2050, other suitable habitats outside of its native range, and a point map of known reported occurrences. They are also combined into interactive biodiversity maps to show patterns of species richness across the oceans and to get a list of species potentially present with a click of an area on the map. Users can also download data directly from the website or request AquaMaps datasets for use in their own work or analyses. These include environmental data layers, georeferenced probabilities of species occurrence, and occurrence data. Large or full datasets can be requested from the AquaMaps project. Since 2009, AquaMaps citations have been steadily rising. Globally, the United States, Canada, Europe, and Australia are the regions where AquaMaps is used the most. AquaMaps has been used in a variety of applications, including marine conservation, fisheries management, climate change impact assessment, ecosystem modeling, invasive species modeling, marine spatial planning, biogeographic and ecological research, as well as education and outreach purposes, demonstrating the potential distribution of species to students, policymakers, and the public. In 2022, AquaMaps began working on AquaX, a new species distribution ensemble modeling workflow that builds on AquaMaps and uses the most recent ecological biogeographical divisions, geostatistics, and environmental variables to predict current and future global distribution of marine species at higher spatial resolutions. This approach uses 10 species distribution algorithms such as generalized linear models, Maxent, random forest, and AquaMaps, and allows for the quantification of model performance and uncertainties for individual species models, as well as macroecological indices such as species richness and change in species rarity. Further model development also involves using expert knowledge to improve modeled distributions. The new AquaX website will be released in 2025. The typical species map page will show the meta-information on the species and their corresponding links to FishBase or SeaLifeBase, a species distribution model selector, the corresponding model mapper, a traffic light indicator on the quality of the map, and an indicator on whether the map has been expert checked or not. In the future, AquaMaps AquaX will further expand its species coverage as it continues to serve as the flagship mapping component of FishBase and SeaLifeBase. The FishBase Guide App, bringing the power of FishBase to mobile phones. FishBase Guide is an Android app launched in November 2021. It is designed to provide producers, consumers, and all fish enthusiasts with the list of all the fish species available in their area or the country of their choice. It also provides the key information on the fish species of their choice, and a store of useful species information relevant to sustainable fisheries management. The FishBase Guide app retrieves data from the wealth of information in fishbase.org. Get the FishBase Guide app from Google Play. Go to bit.ly/fishbaseapp and click the install button to download it on your Android phone for free. The app only requires 15.51 megabytes of storage space, but downloading country information requires additional space. For example, the Philippines, which has 1,035 reported fish species, will require an additional 500 megabytes of storage space. FishBase Guide is a joint effort of Aquatics, Fairfish International, The Sea Around Us, and Mundus Maris, with the goal of raising awareness not to catch, market, or eat juvenile fish that have not yet had the chance to reproduce and contribute to the growth of their population, and appeal to people of all ages on how they can contribute more to the sustainable use of marine and freshwater resources. To start using the FishBase Guide, open the app and select the country of your choice. Or use your location. After you have defined the location, country information will automatically download to your phone. Now, choose the fish you'd like to explore by typing its common name. For example, search for Gigi. The app will display all the fish species with Gigi as their common name. You may filter the search results by common name, family name, or scientific name. You may also sort the search results by size or vulnerability. Click on the specific species box or photo of the fish that interests you. This will bring you to its summary page, which will show you all the information about the particular species you have chosen. Its size, which equates to length, particularly length at first maturity, or the size at which the fish is big enough to reproduce. Optimum length, which is the length at which a fish should be harvested to sustainably produce the maximum eatable catch. And the maximum length, the longest size the species grows or has been observed to grow. You will also find here the red list threat status, which tells you how robust the species is to heavy fishing, climate change, and other stressors. Spawning period indicates the months of the year when spawning takes place. Spawning refers to the process of reproduction where fish release or deposit eggs and sperm into the water. Distribution gives an overview of where in the world the fish occur. More info will bring you to the species summary page on the FishBase website, which provides a more comprehensive set of information about the fish. Applications. Whether you're a fisher, trader, buyer, researcher, or simply a curious fish enthusiast, FishBase Guide is tailor-made for you. It contains the set of essential information. First, to assist you in identifying the species of the fish you have in front of you, even if you only have the common name to work with. It serves as a valuable resource, offering the minimum necessary details for quick identification. Second, to help you become a responsible consumer. The app will let you know if the fish being sold has been caught sustainably, that is, the fish is big enough to have spawned and potentially contributed to replenishing its local population already. And lastly, to help you become a responsible fisher. The app guides you toward following sustainable fishing practices. The vulnerability indicators will tell you what fish can be caught, while the size guides will tell you which fish should be released back into the ocean. So, we invite you to check it out. Get the FishBase Guide app on your Android phone or tablet today. Fish Online presents learning materials for a computer-based study of ichthyology for upper undergraduate and graduate students, using the FishBase information system. It can also be used by independent learners and practitioners in the field of fisheries. On the upper portion of the FishBase search page, just below the FishBase logo, click on Ichthyology course to get to the Fish Online page. The Fish Online Guide is designed to provide structure to a compilation of study materials on various topics in ichthyology. Teachers as well as independent learners will find the materials easy to use or adapt for their learning goals. The topics cover key aspects of ichthyology including evolution and classification, biodiversity and morphology, reproduction, physiology, ecology and fisheries management. The exercises at the end of each section provide learners hands-on experience in locating information in FishBase and doing analyses that illustrate concepts. Teachers of aquatic biology or fisheries can use Fish Online to enrich their lessons with case studies and exercises. Both can be customized to cover species or areas familiar to their students. They can also be customized for a particular focus and learning goal. The exercises and examples can be directly linked to species or thematic information in FishBase, ensuring everyone in the class has access to species and data they are assigned to work on. It is also possible to pick out topics and develop a series of lectures for focused or specialized ichthyology courses. For example, if the teacher wants to focus on the relationships between the size of a fish and where it is at in the food web, a series of lectures can be developed from Section 3.6 Diversity of Sizes. Section 3.11 Diversity of Food and Feeding Habits. And Section 6.2 Trophic Levels and Sizes of Fish. Analyses across all species, or selected sets of species, customized for the student's learning goals can then be made, with data compiled in FishBase. Do you have graduate students exploring topics in ichthyology for possible thesis topics? Graduate students working on their thesis would find some sections of Fish online as good starting points to learn about the state of the science and see research gaps. From there, students can start their exploration of their research topics and write their reviews of literature. Aside from the sections on diversity of diets and feeding habits, diversity of shapes and morphology, biogeography and biodiversity, one can also explore the appendix, which lists topics covered in FishBase. Fish Online can also be used for self-study by practitioners in fisheries management, aquatic conservation, and related fields to understand various concepts in the study of fishes. Let us look at examples of how practitioners can use Fish Online lessons for self-study. Let us look at two sections. The first one titled, What's in a Name?, explaining how fishes get their names. The second, titled, The Length-Weight Relationship, showing how fish weight can be estimated if you have measured its length. Now let's look and see, what's in a name? Indeed, what is the deal with fish species names? Practitioners in fisheries management, aquatic conservation, and related fields often have to write reports containing species names or inventories of species, using a variety of sources of information as references. For those who have not taken graduate-level taxonomy courses, it may be their first encounter with plants or animals referred to by one scientific name in one book and a different scientific name in another book. In such a situation, how would one decide which name to use? The section, What's in a Name?, is a good starting point for a self-study on fish nomenclature, and figuring out what the different ways that scientific names and authors are written in books mean. Go ahead, read that section, and do the exercise using the scientific names of 10 fishes you are familiar with. Include fish with scientific names where the author names are enclosed in round brackets. For example, Tunis albicans where the author name and the year the name was published are enclosed in round brackets. For another example that might be of interest to practitioners, let us look at the section titled, Length-Weight Relationship. In fisheries surveys, fish are often measured for length but rarely weighted individually. The reason is that, in the field, say on a fishing boat that is underway and tossing in the water, it is easier to measure lengths than weigh fish. Often, with the fishing boat tossing in the water, it is just not possible to weigh fish. Assessments and statistics, however, are summarized in terms of biomass or weights. So, the weights of the fish have to be estimated from their lengths. Section 3.7 titled, The Length-Weight Relationship, discusses the concept and provides a step-by-step walkthrough. The exercise at the end of the section shows how weights are estimated for lengths in a length frequency table. In summary, Fish Online presents compiled learning materials for a computer-based study of ichthyology. It is designed for students guided by teachers as well as independent learners. The lessons can be used as is, or adapted for the learner's goals. Examples are provided all throughout, along with directions to where needed information can be found in FishBase. FishBase and SeaLifeBase, premier global information systems providing key data on aquatic species. With extensive coverage, standardized data that is online and open access, and full source attribution, these information systems contribute to efforts addressing global challenges such as access to information, biodiversity loss, habitat degradation, food security, and climate change. And likewise, to achieving global policy targets such as the United Nations Sustainable Development Goals toward protecting and conserving life below water, as well as ending all forms of malnutrition. These have been made possible through the invaluable support of partners and donors throughout the years. and through the dedication of members of the FishBase and SeaLifeBase teams, and the FishBase Consortium.